All right. So a second advantage to organelles, membrane-bound organelles, mind you, is that cells can confine potentially harmful proteins and new molecules in membrane-bound organelles and thereby protect the rest of the cell from those contents. So a cell can concentrate the substances within an organelle, increasing rates of reaction, remembering again that increasing enzyme concentration will increase rate of reaction. Increasing substrate concentration up to a point will increase rate of reaction. And increasing, but also if you increase harmful enzymes, if you increase the concentration of harmful enzymes, or you produce harmful enzymes that can break down other proteins, then you will have a problem in a cell that is made up of proteins that have a job to do. So you need to keep those things that can cause problems in other parts of the cell away from the other parts of the cell. So wall off the stuff you don't want to cause problems, but you still need. Now, remember this slide? It wasn't that long ago. Well, these are the organelles themselves. Let's talk about those individual organelles one at a time. Now, this is a big slide, and I don't really want to talk about it using this slide. And this is the plant cell using this slide. I'd rather do the thing that I made. Yes, I know, this is terrible. But this is a recap of those slides. And effectively, we have our lipid bilayer. So our lipid bilayer is this image down here. Our lipid bilayer is composed of our phospholipids. Our phospholipids with their hydrophilic head and their hydrophobic fatty acid tails in a bilayer structure. One head, two tails, in the tails interacting here and the heads interacting here. Hydrophilic substances or solvent out here, hydrophilic solvent out here, hydrophobic core here. Now, other things composed in there, there's a range in size of 5 to 9 nanometers. This lipid bilayer surrounds the cell. This is the cell's surface membrane. We also have the nucleus and the nucleolus. The nucleus contains DNA. The nucleus is the site of transcription of RNA. RNA is transcribed from DNA. Now, our RNA is specifically transcribed in the nucleolus. The nucleolus is those dense regions, those spots. This spot right here, that's the nucleolus. That spot right there, that's the nucleolus. Now, I want you to understand something. There can be multiple nucleoli. There could be more than one. A single nucleolus, multiple nucleoli. In a plasma cell, for example, which we will get into in much greater detail in units 4 and 5, those will have multiple nucleoli because they need to produce lots and lots and lots and lots and lots of ribosomes. So you need lots of rRNA. The nuclear envelope. The nuclear envelope is a double membrane, similar in structure chemically to the cell surface membrane, but two cell surface membranes. So you've got membrane, lip, membrane. So that double membrane is going to enclose and separate the nucleus of the cell, but it allows the passage of RNA through the nuclear pores. Now, ribosomes are these organelles. They are not membrane-bound organelles, but they are structures that are important for the metabolic processes of the cell. They're combined rRNA and protein. They will be the site of translation, the production of proteins, the synthesis of proteins, polypeptide chains. They are found in eukaryotic cytoplasm as ADS ribosomes. They are also found attached to rough endoplasmic reticulum, but they are still on the cytosol side. Now, they can also be found in prokaryotes as the 70S ribosome size. Mitochondria and chloroplasts have their own ribosomes. They are 70S ribosomes. So do not be fooled by questions that ask you, what kinds of ribosomes do you find in a eukaryote? 80S, 70S, all of the above? The answer is all of the above, because you can find mitochondria and chloroplasts in a eukaryote, and they have 70S ribosomes. And you got cytoplasm in a eukaryote, so you got 80S. Understandable? I hope so. So, let's move on to your smooth endoplasmic reticulum. Your smooth ER, SER for short, but you should spell out the whole word on the exam. These are a system of interconnected internal membranes that are connected to the nuclear envelope. They play a role in the fabrication of carbohydrates and lipids for the cell. Now, the actual internal network is called a network of cisternae. Cisternae are the outpouchings and cavities, the dead ends. Cisternae. You can get extra points for describing that terminology. That spelling too. C-I-S-T-E-R-N-A-E. So, those are the cavities. Now, rough endoplasmic reticulum in structure are similar to the smooth endoplasmic reticulum. However, they are made rough by the fact that they have ribosomes attached to the exterior surface, the cytosol side. Now these ribosomes that are attached to the rough endoplasmic reticulum, these are 80S ribosomes. These rough endoplasmic reticulum, this organelle's express purpose is for the fabrication of proteins for the cell. It has that job. That is the main purpose, and that's what you write. Now, Golgi apparatus. That's my preferred way of saying it. However, Golgi body is shorter to write. You're less likely to spell it wrong. So, Golgi body for now. It is a series of separated, stacked membranous structures that serve to collect, modify, package, and secrete cellular products. Lysosomes. Lysosomes are vesicles that contain digestive enzymes for the breakdown of macromolecules and old damaged cell parts. Lysosomes are made by the Golgi apparatus. Excuse me, Golgi body. I'm going to make that mistake for the rest of my life. Now, peroxisomes. Peroxisomes are vesicles that contain enzymes and antioxidant molecules for the reduction of reactive oxygen species. So hydrogen peroxide especially. They also break down very long fatty chain. They also break down very long chain fatty acids through beta oxidation. So this is important for fueling mitochondria in the process of aerobic respiration, and also for the precursor production of lipids for lipid molecules for use in the plasma membrane. So they've got a number of different things. A number of different functions. However, where do they come from? Well, they're vesicles containing enzymes. Well, they're probably going to arise from the Golgi body. Didn't make the mistake this time. I'm learning. All right. So here's a transmission electron microscope image. These grill marks right here, this is rough endoplasmic reticulum. We can see the ribosomes studding the membranes. I have a nucleus here. I have another nucleus here. It's roughly round. It's relatively circular in shape, but I only see half of it right now. I can see heterochromatin and euchromatin. We'll discuss that more later. And I can see here mitochondria, 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 mitochondria. These mitochondria are recognizable because they have these inner membrane folds called cristae. Now, cristae are, did I write them down here? I did not. All right. So, mitochondria have cristae, which are inner folds. And those inner folds is where the electron transport chain is going to occur. And that's more of an A-level topic, according to the 2016 to 2018 curriculum of Cambridge, so we're not going to cover that in these videos. Now here I have an open space. That indicates to me that I have a cell membrane running along this path. But I'm going to close up on that cell membrane. Remember that I can get good resolution and really good magnification using transmission electron microscopes. I can see cytosol here, some kind of vesicle over here, and I can see these tram track appearance. This is a cell membrane. I see a hydrophilic head in black, a white space in here that is the nonpolar fatty acid tails, and another black line here that is the hydrophilic head. There's also an intercellular space, the space between two cells, and another tram track right there with a hydrophilic head, hydrophobic tails, hydrophilic head. So I can see the lipid bilayer. This is somewhere between 5 and 9 nanometers in diameter, right there. So that's my scale. That's the kind of magnification resolution I can get with these images. So, now let's talk about the nucleus structure. I have a nucleus, the full round nucleus right here. I've got the double membrane running all along here. I've got euchromatin, the white patches here. I've got heterochromatin ringing the entire rim of my nucleus. I've got a nucleolus here and a nucleolus here. And these nucleoli, they don't have to be perfectly round. They also don't have to be just one. Here, I can see a transmission electron microscope, another one, of my double membrane. I see a membrane here. I see a membrane here. Here is my nuclear pore, right there. That's where my RNA is going to escape. Here I've got a mitochondria with the cristae folds in there. Now, I wonder if you can spot the nucleolus right here. Yeah, that's a nucleolus. This right here is the nucleus. That is a double membrane. All this is euchromatin. This is cytosol. This is a cell wall, indicating to me that this is a plant cell. Now, this is the process of protein production, protein export. DNA in the nucleus is transcribed to produce RNA. mRNA is excreted through the nuclear pore and moves into the cytosol. It is clamped down upon on the small and large ribosomal subunit, Mr. Clampy, if you recall. And that will either begin the process as a free-floating polypeptide, or that ribosome can be attached to rough endoplasmic reticulum, and that polypeptide can then be formed into the endoplasmic reticulum space. Now, if the endoplasmic reticulum space, that polypeptide is excreted into here, it will go through this process, and there will be some post-translational modification occurring as this moves, and it will be moved to a cisternae, and then a vesicle will be pinched off. A endoplasmic reticular vessel will be pinched off, and it will be carried to the Golgi apparatus, Golgi body. Once there, it will be packaged, processed, shipped, packed with other molecules like it, or similar necessary molecules. It will move from layer to layer to layer, picking up new additions as it needs to, and then it will be shipped out. If it is tagged to be secreted from the cell, then it will be moved to the cell membrane and exocytosis will occur. If it needs to become part of a lysosome, it will be packaged with other lysosomal products. If it is tagged to become a peroxisome, it will be packaged with other peroxisomal products and excreted that way. So depending on the necessary fate of that protein, that's what the Golgi will do. That's what the Golgi body will do. Now, here's another one of those tables from me. Centrioles are unique to animals. They are a pair of complex bundles of microtubules. They assist in the cellular process of cellular division. They are not, by any means, required for cellular division. They are assisting. If you took the centrioles out of an animal cell would still be able to undergo cellular division. It is still possible. They are just better at it with the centrioles than without. Plant cells that do not have centrioles can still divide. It's possible. Now, microtubules. These are protein tubes. Form the basis for centrioles, cilia, flagella. They also act as part of the cytoskeleton. Cytoplasm is a semi-fluid matrix that contains the cellular organelles. A cytoskeleton consists of actin filaments, intermediate filaments, and microtubules. They hold organelles in place and allow the movement in certain types of cells. Mitochondria, they are a double membrane-bound organelle. They have their own circular DNA, which is abbreviated mtDNA. It stands for mitochondrial DNA, mtDNA. I'm going to spell that out for you, just in case you can't hear it in my voice. Mitochondrial DNA. I hope that's visible, even though I wrote it in green. mtDNA. So, they are tasked with the production of ATP, and lots of it. They produce ATP. I'm going to give you 3 rules. First rule of mitochondria, make ATP. Second rule of mitochondria, make ATP. Third rule of mitochondria, make more mitochondria so you can make more ATP. So, the organelle that houses the chlorophyll, those are the chloroplasts. The chlorophyll is housed in, the chlorophyll pigment is housed in thylakoid membrane bundles called granum. They have a key role in the photosynthetic production of carbohydrates. Chloroplasts, much like mitochondria, they also have their own circular DNA, which are called ctDNA, which stands for chloroplastic DNA. Chloroplastic DNA. Because they couldn't be bothered to come up with something better like cpDNA or something, because that would fit so much better. But chloroplastic DNA, we got ctDNA. It's what we got. We're going to live with it. All right. So MT for mitochondrial, chloroplastic, ctDNA. All right. Now, chloroplasts also have 70S ribosomes. They also divide on their own. They will also produce ATP. But